Hello, welcome to the next A-level lesson from the conscientious biologist, Ben Gallagher. This one's the second one in the Saccharide Biochemistry set. This is on polysaccharide structures and functions. The last one looked at glucose biochemistry and glycosidic bonding. It also gave you a bit of an overview. At the end of this video, we're going to revisit that overview to complete all the ones that were left blank at that stage. As I've said, this is A-level syllabus, and please do subscribe to my YouTube channel if you haven't done so already. And don't forget, I put lots of supplementary information on both Facebook and Instagram. Thank you. So let's do a very quick recap of glycosidic bonding, because you'll need to remember both forms, the 1,4 glycosidic bonds and the 1,6 glycosidic links, if you're going to know how to properly apply them to polysaccharide biochemistry. So if I put one alpha glucose molecule up there, which in the last video I took you through how to draw that and how to remember it, go back and watch that if you're not sure. If I put a second one next to it, remember it's those two OH groups of adjacent alpha glucose molecules that do the glycosidic bond, because they lose 2 hydrogens and an oxygen, so that's H2O, because it's a condensation reaction that leaves an oxygen in the middle to form the oxygen bridge, which I'll mark there as the glycosidic bond. Now, because that's between the first carbon, the first one after the oxygen as you're going around clockwise, between the first and the fourth, the one at the back of the nearer alpha glucose, that's a 1,4 glycosidic bond between the first carbon and the fourth carbon. A 1,6 glycosidic bond is if you get the OH group above the 6th carbon, so on the mouse's tail, if you remember that. If the OH on there becomes close to the OH group on the first carbon, those two that I've circled here, they can lose H2O. So again, it's a condensation reaction. The oxygen is left over to give you that 1,6 glycosidic bond. So a glycosidic bond forms between two molecules, two glucose molecules, through a condensation reaction between OH groups on adjacent molecules, giving off water as it forms. That's your perfect description of a glycosidic bond formation, including the keywords, but make sure you can recognize them and make sure you know the difference between the two bond types. So let's look at our first polysaccharide now. This is amylose. So what I'm going to do is pull up another alpha glucose molecule there, slightly smaller, but instead of drawing out in full when we do polysaccharides, because it's too much hassle drawing out a full complex every atom when you've got to draw many of them. So we're going to substitute that with a simpler diagram, which you may remember from my building up alpha glucose in stages as kind of the first step, drawing the mouse. So he's got his tail and his eye, but that's representative and a fairly well recognized shorthand diagram of a glucose molecule. Now in amylose, if you take another alpha glucose and put it in front, you get your glycosidic bond formation, which I've drawn on there. So you can see I've kept it in brown to make it obvious. But if you keep getting alpha glucose molecules joining at the front and the back, what you get is this kind of long straightish chain. That's an amylose polysaccharide. Obviously much longer than this, so I'll just pop an etc. above me here. But these very, very long chains of alpha glucose molecules, that's amylose. Amylose is one of the key polysaccharides that you've got in starch. The other key polysaccharide that you have in starch is something called amylopectin. Okay, so if I change the title there to amylopectin, and amylopectin, not dissimilar, it's still alpha glucose, but it also has 1,6 glycosidic bonds. You can see I'm just putting a couple up there, so you can see I've got a branch coming out of it. If I remove these, look, I could branch downwards, I could go backwards, I can go forwards, I can come across. Depending where you put things, you can combine 1,4 and 1,6 glycosidic bonds to make a much more highly branched structure. Now that's amylopectin. Now for amylose and amylopectin, we're going to put a couple of key points up above me here. So amylopectin contains both 1,4 and 1,6 glycosidic bonds. So it's highly branched and very compact. Amylose, because it's longer and straighter, it's not as compact. Okay, so amylopectin is really good. Now, like amylose though, amylopectin is insoluble, so it has no effect on osmosis. So it's the perfect storage molecule, and amylose and amylopectin together form starch. Perfect way for plant cells to store their glucose. Think about osmosis, think about concentration gradients. If they just put the glucose molecules into their cytoplasm, it would lower the water concentration, the water potential. So water could move into the cell to try and dilute the glucose, make the cell extra turgid, put it at risk of bursting, certainly mess up with the movements of other things going on. Whereas converting alpha glucose, which is what's produced by photosynthesis, polymerizing it, sticking them all together into amylose and amylopectin, makes it small, compact, insoluble, so it doesn't affect osmosis. It's the perfect storage molecule. Some starch, as I said, is a mixture of amylose and amylopectin. So make sure you know the differences there. Key one, amylose, just 1,4 glycosidic bonds, amylopectin, a mixture of both. Now, if we change this from plants to animals, we can change the title again to Glycogen. I'm going to get rid of the text above me now and change this to glycogen, because glycogen is essentially the same as amylopectin. Look, I haven't changed the diagram. It contains both 1,4 and 1,6 glycosidic bonds. But, and what I haven't shown in my diagram, but I will do on a summary one in a couple of minutes, glycogen is a much larger molecule and even more highly branched. Okay, glycogen is what we animals store our glucose as glycogen, whereas plants store it as amylose and amylopectin as starch. Okay, so pretty quick run through there because it doesn't often come up in the exams. When it does, it's going to be recall of key facts describing the differences. They might give you a table with amylose, amylopectin and glycogen and ask for the key differences. It's largely about their bonding, their size, how compact they are, but all of them are insoluble, so all of them are perfect storage molecules. Moving on then, let's look at cellulose and beta glucose. We've done lots on alpha glucose. Everything so far has been drawing the structure of alpha glucose and how alpha glucose polymerizes, how alpha glucose makes its glycosidic bonds. But in that first summary page on the previous presentation, we had an overview that included beta glucose. So what I'm going to do, I'm going to pull up alpha glucose here in this box above me, and I'm going to turn alpha glucose into beta glucose because it's really simple. You just need to focus on the front carbon, so that's carbon 1, and the H and OH groups there, because it's really simple. Watch as I change it, okay? You get rid of them, you bring them back the other way up. It's that straightforward, okay? The only difference between alpha and beta glucose is that the front H and OH groups are the other way up. Now, why that's really important, because it impacts how it arranges itself when it forms glycosidic bonds. So to show you, got the alpha glucose molecule over there. If I pull up another alpha glucose next to beta glucose, sorry, if I pull up another beta glucose next to it, notice something about the beta glucose. To get those two OH groups side by side, I've had to put the second beta glucose upside down because the OH on the front of that first one is now up high, but the one on the back is still down low. So to get them aligned, I have to flip it over the other way. So now that that second one's upside down, the OH groups can form a glycosidic bond. That glycosidic bond will pair those two together. Think about now if I have to add a third one at the front. The third one can go back to being the right way up again because the middle one was upside down, meaning its OH group at the front, which is up high when it's round the right way, but because it's upside down is now down the bottom, which is where the OH group on the fourth carbon is on the new one. It all gets a bit confusing, but hopefully this is making sense. They've got to alternate so that those OH groups are now down the bottom. So look at what we've got. We've got one that way, the next one flips, the next one flips back, and they would just continue to do that. So if I put a whole chain of them down the bottom, you can see that they just alternate, forming the glycosidic bonds between them. So cellulose is made of long polymers of beta glucose. Those long polymers, though, then form cross-links by hydrogen bonding to other long strands of cellulose, which means they can form a really good mesh. I'll show you a diagram of that in a slide in a second. But you must, in the exam, be able to describe or draw the differences between alpha and beta glucose, which is really simple because it's just the difference on that front carbon. But you've got to be able to describe the difference. Another good thing as well, the reason why it does that, or perhaps a useful outcome of it doing this, is it makes them form very straight chains. The chains that you make with amylose, which if you remember amylose, they're all in a straight line like cellulose, but in amylose, it's alpha glucose, so they all go the right way up. Amylose has a tendency to bend and turn, so it forms big kind of spirals. Whereas cellulose forms long straight chains because of that alternation. Never gets a chance to spiral because as it starts to, it goes the other way up, takes it the other way. Okay, so cellulose, really good long chains of carbohydrates, great of course for making plant cell walls. So if we look at a summary of these diagrams, if we start off with the two that are in starch, so we've got amylose, that's these long chains of alpha glucose. So it's alpha glucose polymer of 1,4-glycosidic bonds. Remember what I just said a minute ago, that does have a tendency to form spirals. If you look at amylopectin, it's still alpha glucose, but now it's branched because it contains 1,4 and 1,6 glycosidic bonds. Glycogen is the same as amylopectin, but much bigger and much more highly branched, which you can see on this diagram. But it's still, look at the descriptions, they're the same. It's still a branched alpha glucose polymer of 1,4 and 1,6 glycosidic bonds. It's just glycogen is much bigger. Contrasting that with the last one then, with cellulose, cellulose is these much straighter chains. It's beta glucose polymers, 1,4 glycosidic bonds, but remember they can make these cross links with other straight chains to form this much better kind of mesh that's perfect material for building plant cell walls. Going back to the first slide of the previous presentation on saccharide biochemistry then, this was the first slide. We're now going to fill in the blanks. Remember, saccharides are just sugars, more commonly known as carbohydrates. Things like this, natural ones over the far side, things that you get in fruits and potatoes, vegetables, bread, pasta, which are a bit more processed, right down to really highly refined sugars like sugar cubes in the middle, and then sweets, chocolate, cakes, things like that. But remember all your monosaccharides, so alpha glucose, you now know its detailed structure and can draw it, and that's the main respiratory substrate. It's what we break down in respiration to get the energy out of it. But beta glucose we can now also add to this. We know that beta glucose is used to make cellulose, and you need to be able to draw both alpha and beta glucose. Deoxyribose and ribose, both part of DNA. Fructose and galactose, both isomers of glucose, both have the formula C6H12O6, exactly the same as glucose. They're just slightly different versions. Can be used as respiratory substrates, but more commonly they're converted in the cell to alpha glucose, and the alpha glucose is then respired. Disaccharides, this hasn't changed because it was complete when we looked at it in the previous video. Disaccharides are where you've got two monosaccharides bonded together. The three key ones are maltose, sucrose and lactose, and you need to know what they're made of. Maltose is the most common one because it's just two alpha glucose molecules stuck together by a 1,4 glycosidic bond. Sucrose is alpha glucose and fructose, fructose which we had over there as a monosaccharide, and lactose, which some people are intolerant of, is alpha glucose plus galactose, which was the last monosaccharide over there. Polysaccharides are what we've been looking at in this video though, which is when you've got lots of monosaccharides bonded together. Amylose we know is alpha glucose polymer for storage in plants. I haven't put amylopectin on here because it's amylose that's more important for you to know in your syllabus. But please remember that it's amylose and amylopectin that both form starch. Okay. Glycogen is a more highly branched polysaccharide. It's still alpha glucose, but it's how we store our glucose in animal cells. Cellulose we know is made from beta glucose. It's used for plant cell walls because you can form these big meshes. Chitin is used in fungal cell walls, and peptidoglycanol murane should ring a bell from when we did our prokaryotic biochemistry because it's the key intrinsic part of bacterial cell walls. So this video covers everything then that you need on polysaccharide biochemistry. So go back, take any screenshots that you need, but you do need to be able to describe these structures and relate it to their functions. So we've now covered all of your biochemistry. Hopefully you've already watched the videos on DNA biochemistry, lipid biochemistry, which was triglycerides and phospholipids, amino acid biochemistry, which was the start of protein synthesis, and now we've done all your saccharide biochemistry. That's a huge amount of the A-level course. From here, head to meiosis 1. I think meiosis and cell division is probably a good place to move on to next. You should have already studied mitosis in one of my videos. This is now the form of cell division that creates variation, creates gametes to allow for sexual reproduction. So head to that one next. Please do give this video a thumbs up if you found it useful. Do subscribe to my YouTube channel if you haven't done so already. And remember to follow all the extra information that I put on Facebook and Instagram as The Conscientious Biologist. Thank you.